Good morning. Last lesson was really the first lesson of the nervous system. We kind of just gave some general functions. But at the end of it, we went into a little bit of the structure of a neuron. Neuron is kind of like the fundamental unit of our nervous system. So today we're really going to go into how neurons actually function. What do they do? When your brain is functioning, when your muscles are moving and you're relaying information, really it's just kind of a relay of signals. It's like playing telephone, right? Relaying a signal from one region to another region. And how neurons relay messages is they relay messages through the neuron with an electrical impulse. And then they relay that message to the next neuron using chemicals known as neurotransmitters. So we're going to start today kind of talking about like how neurons generate that electrical impulse and how they can fire, how that impulse starts. Okay. Neurons are maintained in a state known as resting potential. Now, resting potential is kind of sounds a little backwards. Resting potential is a charged state or a polarized state. So polarized charge are kind of going hand in hand right now. So when neurons are in resting potential, they are in a charged state. I like to think of the concept of pulling back a bow and arrow. So if I pull the arrow back on the bow and I hold it in this position, it has a lot of potential energy. And it's resting in this position, getting ready to fire. So when a neuron's resting potential, it is charged, it is polarized. When the neuron fires and sends this electrical impulse, that is known as action potential. Okay. Some of the key concepts we're going to kind of use to talk about how neurons function. We're going to think about concepts of concentration gradients. Going to think about diffusion through the membrane with passive transport through like transport proteins. We're also going to talk a lot about active transport with the sodium-potassium pump. You probably remember that way back when. So looking at this here, sodium-potassium pumps play a role with help like maintaining that resting potential, right? And when we talk about this resting potential in this charged state, we're talking about electricity. So we're talking about ions, the movement of ions, and that's what neurons do is they move ions around. So if we look at this, here's kind of just a cross section of a neuron, right? And it's happen to be at the dendrite section, but it could be in the axon. Fairly similar for our purposes right now. And you can see we have a high concentration of sodium outside the cell and a low concentration of sodium inside the cell. So if you think of the sodium-potassium pumps, potassium pumps can pump sodium out, so creates a high concentration of sodium out of the cell, and then they pump potassium in. And you can see this here, that there's a high concentration of potassium inside the cell, low concentration outside the cell. Now, what's weird about this, if you're kind of looking at it, like if I'm moving both positive sodiums and positive potassiums, how am I creating an electrical difference, a potential difference? Well, the other kind of side note here is we have these very, very large negatively charged proteins in here. So kind of symbol right here. So negatively charged proteins in there. And those proteins help to kind of maintain the negative charge inside. So in any case, with the sodium potassium, what they're doing is we're creating a positively charged region outside the cell and a negatively charged region inside the cell. And that is resting potential. Now, if you think of nature doesn't like to be out of equilibrium. So when you create this concentration gradient, this electrochemical gradient, there's energy stored there, right? There's energy of all those ions wanting to get back to equilibrium. Kind of a little bit like the concept, if you shuffle your feet, like on a dry day in the winter and you shuffle your feet on the carpet, and when you're shuffling your feet, you're picking up extra electrons, right? And so you're becoming charged in a sense. If you go to touch the doorknob, right, all those extra electrons want to get back to equilibrium. So what happens when you touch the doorknob is that you have a little shock, and that's the electrons jumping back, right, trying to get back to equilibrium. So essentially what we're doing here is we're creating this electrical charge across the membrane, creating this potential difference. Okay. So with all cells basically have some type of charge across their membrane. Neurons have a cell that's typically about negative 70 millivolts. And that's just a good number to know. It's just the amount of charge across the membrane. You guys know physics, right? The negative just tells you direction. So it depends on which way you're measuring it, but we measure it in a certain way that it comes to negative 70 millivolts. That's a good number to know. And really what helps to kind of maintain this charge is a sodium-potassium pump. Okay, looking a little bit more in detail here, right? We now can see our sodium potassium pump, pumping the sodium's out, the potassium's in. We also can see here these arrows. These arrows are kind of demonstrating or showing passive transport channels, right? Transport channels can be very specific though. So this is a transport channel that allows for the movement of sodium. Here's another different protein that's allowing for the movement of potassium, and this is passive transport. So now you can kind of get a better picture of how we're maintaining this potential difference, right? See how the sodium arrow is small? It's only allowing a very small, slow, almost no trickle of sodium back into the cell because I'm pumping that sodium out, get the positive on the outside because I want the positives on the outside, negative on the inside. You kind of see this potassium one's opened up a little bit more. So even though I'm pumping these potassiums in, it's kind of always kind of slowly trickling out and helping to kind of maintain this balance of potential difference here, right? So again, we are charged positively on the outside and negative on the inside, and we are in resting potential right now. Okay. Now, how do we get a neuron to go from resting potential to action potential? Is you need a stimulus, right? A stimulus could be a little heat sensation or touching. We're going to think of stimulus in terms of neurons first, right? In terms of neurons stimulating another neuron with neurotransmitters. So you kind of ignore this first one here. But if we look at this, here is a stimulus. Here's my resting potential. And there's some type of stimulus causing some change or some depolarization, right? Because it's polarized here. It's getting slightly depolarized. But that stimulus isn't strong enough to really get neurons to fire. Neurons either fire completely or they don't fire at all. So right here, there's no firing between the neurons. Okay. Now, if you look on the right-hand side, right, you can see that we had this stimulus that was just strong enough to get to this threshold, which is like a change of negative 15 millivolts. And you can see this stimulus is strong enough to reach kind of this launching point, this threshold, and then the action potential takes place. Okay. So stimulus is kind of the key to starting an impulse. Okay. So looking at here, we're looking at a little bit more now. We really have a whole bunch of protein channels that play a role here. So here's all these sodium and potassium channels that were showed a couple slides ago, right? And they're lining the axon. They're lining all the membranes of your neuron. And they're typically in a closed state, right? Not really letting anything through because we're trying to maintain this potential difference of resting potential, this polarization. However, when a stimulus hits, a stimulus can cause the sodium and the potassium channels to open up. And if the sodium potassium channels completely open up, you can see what happens. All the charges try to get back to equilibrium. So therefore, all the sodiums will start rushing into the cell and the potassiums will start rushing out of the cell. And that is like the electrical impulse. That is the action potential. Now, when your neurons do this, all of the sodium and potassium channels don't open up all at once. They kind of propagate like a wave down the neuron. So you can kind of think of if you went to a sports game and you see people doing a wave in the stands, one section stands up and then sits down, then the next section stands up. You can kind of see the wave propagate around the stadium. That's kind of how this happens, right? These channels here open up, allowing the impulse to take place in this region. They immediately close down, and then the next section opens up, close down. The next section opens up, close down. Next section opens up, close down. So this impulse propagates like a wave down the neuron. Okay. So looking at this just a little bit more is now we can kind of combine the two actions here. So here is kind of measuring the polarization and depolarization of our impulse. Here's what's actually going on with the sodium potassium channels. So you can see here in stage 1, I am in resting potential. So I am in resting potential right here. And hold on one second. In resting potential right here, you can see that we're maintaining our charge. Our sodium potassium pumps aren't shown, but they're working, right? And we have a high concentration of sodium outside the cell, a concentration of potassium inside the cell. All right. Right now we're resting. We're just maintaining charge. So therefore, your kind of sodium potassium channels for the most part are just closed up. Okay. I have a stimulus, right? The stimulus, right? Neurotransmitters, something stimulates the neuron. The stimulus hits the threshold. And what happens when that stimulus hits the threshold? And as it's reaching the threshold, its stimulus is actually causing the sodium channels to open up in one region. And if it's strong enough, you get enough of these sodium channels to open up that it really causes this action potential. And you can see here, I'm going to move this over here, with this action potential, you can see what's going on is all these sodium channels are open up. And if these sodium channels open up in 2 and 3, here's 2 of them starting and then really opening, all those sodiums rush into the cell. Well, think about what happens. The cell is negatively charged inside, positive outside. Very, very polarized. When all the sodiums rush into the cell, you could see rushing to equilibrium, to being neutral. In fact, so many sodiums rush into the cell, they actually kind of flip the charge. So this process is all the positive sodiums rushing in, and that's called depolarization. That's action potential. I'm very much depolarizing and even going past neutral, past equilibrium of that neuron. And that's that electrical charge traveling down the neuron. Now, as soon as a neuron fires, they need to kind of repolarize. So the next thing that happens is all these sodium channels will then close up here, and then all the potassium channels open up. And now those potassiums are reversed. So they're high concentration inside, low concentration outside. So they immediately rush out of the cell. And they're positive charges as well. So when they rush out of the cell, it's causing that cell to become more and more negative. So this is repolarization. Typically when this happens, it overshoots, right? So this is kind of the overshoot right here, hyperpolarization. And this is kind of a period of time where the neuron can't fire. It's just kind of this like refractory period until it gets back to like stable resting potential. Okay, so that's how a neuron fires in terms of sending its electrical impulse, but that's not really how neurons communicate with each other. They don't really send electrical impulses from one neuron to the next. This is all contained in just one neuron. So the next part is to kind of look at, well, how do neurons actually communicate between each other? Well, we kind of showed it before, right? Here's my neuron, right? The connection between neurons is called the synapse. Okay, here's my synapse right here, right? So this is my presynaptic neuron. This is my postsynaptic neuron. And here's the axon, and these are the axon terminal buttons. So this is the axon terminal button, and it's in close proximity, very, very close proximity to the postsynaptic neuron. And this little cleft right here is called the synaptic cleft. Now, with this, how neurons communicate with each other is with a chemical known as neurotransmitters. There's a lot of different neurotransmitters out there. Dopamine, serotonin, right? A lot of different types of neurotransmitters out there. We're not really going to talk specifically about them. We're just going to talk about them in general. So what happens with this is this action potential propagates all the way down to the axon terminal button. And what it causes is it causes neurotransmitters to be released into the synapse. Now, inside the axon terminal buttons, there's a lot of neurotransmitters being produced. They're being stored in vesicles. Those vesicles will connect to the membrane. Exocytosis takes place and releases all of these neurotransmitters. What those neurotransmitters do, they're just chemicals, right? They're chemicals that are going to bind. So they're like a ligand. They diffuse across the synapse, and I have all these receptors on the other side. And these are protein channels that have receptors on them. And what they do is the neurotransmitter matches up with the receptor. And when the neurotransmitter binds to the receptor, it causes the protein channel to open up. And that protein channel could be an ion-gated protein channel, again, allowing like sodium or potassium to go through. I'm going to kind of go to the next slide here. All right, so looking at it a little bit closer and a slightly better one, okay, really what happens here is this. Right, that impulse propagates all the way down. The electrical impulse gets down here. It actually causes the electrical impulse actually causes this calcium channel to open up. And when that happens, calcium rushes into the neuron, and that calcium rushing into the neuron is what causes the release of the neurotransmitters. This is all in the presynaptic neuron. Now, those neurotransmitters will be released. They'll diffuse across the synapse because it's a very, very small space. They'll bind to the receptors on the ligand-gated ion channels. Ligand is anything that binds to a receptor. Gated channel means I can open and close it. And what am I letting through? Ions. Okay. So they bind here and they open up the channels. This is where you can kind of see the difference when I showed here in terms of having a stimulus that's strong enough here that reaches a threshold or one that's not strong enough to cause action potential here. And this is, you can actually see it here with this image. Now, let's just say like one neurotransmitter is released or a couple neurotransmitters released. I only open up one of these channels. There might be a little kind of tick in terms of change in polarity, but it might not be a large enough stimulus to cause the entire neuron to fire. Maybe all of these channels need to be opened up for that neuron to fire. Okay, so now as soon as this happens, right, the neurotransmitters bind to the receptors of the ligand beta ion channels. They open up and all the sodium potassium starts rushing. As soon as the sodium potassium start rushing, that's where the impulse starts to propagate throughout the entire neuron, and that next neuron's going to fire. Just an interesting thing about neurons, like obviously, if you touch somebody, you kind of just tap somebody, or if you punch somebody, or if you have a little pinprick, something like that, you can kind of sense different amounts of pain and pressure. It's not how neurons fire. All neurons either fire completely or they don't. So it's not like when you feel something really, it's a strong stimulus, like something really hurts. It's not like the neurons fire harder. They don't, they just fire. The difference is it's the amount of neurons and the rate at which the neurons are firing. It's like the density of neurons that are firing, and the rate at which they're firing is what causes something to have a greater stimulus, more pain versus less pain. Okay. One last kind of couple things here. Last couple things here is we kind of mentioned the myelin sheath and how it kind of plays a role with speeding up these electrical impulses. And now we can kind of talk about it a little bit more. So if a neuron is unmyelinated, you need to depolarize and repolarize every inch of that neuron, every inch of that axon to reach the end, the axon terminal buttons for that for the impulse to be transmitted via neurotransmitters to the postsynaptic neuron. However, if it's myelinated, the myelin sheaths act as conductive material, and what happens there is these electrical charges, these ions, the charge can jump through the myelin sheath. So therefore, I don't have to depolarize the entire neuron. I just need to depolarize and repolarize the spaces in between the neurons. So therefore, I am depolarizing and repolarizing far less of the neuron, which makes that impulse go drastically faster, exponentially faster. So we call this the myelin sheath, and the spaces in between that we actually depolarize are called the nodes of Ranvier. Nodes like a kind of in between the two spaces. So these are the nodes of Ranvier. Ranvier is the one who discovered the nodes. Interesting thing about the myelin sheaths is they're produced from cells known as Schwann cells. Theodore Schwann was the one who discovered it. Theodore Schwann, cell theory, right? All animals are made of cells. So you kind of like two things going on in our book. Any case, so that causes this to kind of propagate down, and the myelin sheaths play a role. If you have something that's destroying your myelin sheaths, it can have severe consequences. One last thing, just kind of looking at, this is a good graph to know, right? We have our resting potential. Right? We have our stimulus that sometimes if it's not strong enough, the neuron doesn't fire. If it's strong enough, it hits the threshold. Depolarization is the opening of all the sodium channels. Then they close. Repolarization is the opening of all the potassium channels. Then they close. They tend to overshoot, become hyperpolarized until they get back to their resting potential. That's it. That's how a neuron fires. Hopefully you found it interesting. And if you have any questions, we'll talk in class. Thank you very much.